Today we'll talk about the genetics of 6, 7. So stay tuned. Elif, Malak, and Rebecca. I'm so happy that you guys decided to ace this unit. So keep up the good work. Go girls. Have you ever looked in the mirror and thought, why did I get my mom's nose? My dad's eyes? Well, as you all know, that relatives look alike. Like this girl looks like her mom, this daughter looks like her dad, and these two boys look like their dad. So that's an observation throughout history that we've been seeing, that kids look like their parents. And the same applies to certain disease. So certain disease run in families. So one disease such as electrodactyly, in which the hand would look a little strange, runs in families. And one family is the Stiles family. You could see that Grady here has that disease, and many of his relatives. Relatives look alike everywhere. If you look at the Roya family, they have that Habsburg job. You could see it from generation to generation. We also see it in the animal kingdom. You could see animals look like one another. So people throughout history have been asking, why do relatives look alike? Many people have been asking that question. And one specific person was Gregor Mendel. And he lived in the 1800s, and he asked that question. Why do relatives look alike? And how are traits passed from parents to offspring? And he was thinking, well, for this month, my assignment is to figure out exactly how traits are passed from parents to offspring. I'm going to discover this. I'm going to be famous. Kids will study about me later in life, right? He was thinking that. So, but there's a problem. He has a problem. He needs to decide what type of model is he going to use, right? So if you wanted to cross two different individuals and look at the progeny, you better pick something that doesn't take so long. The cats take about 2 months to produce their offspring, sheep 5 months, and humans like 9 months. If you were Mendel, what would you pick? Cats, sheep, or humans? Well, none. He picked none of these. He actually went with something different. He actually picked peas. And peas, it takes only 60 days. Well, why not cats, you may ask? If it takes 60 days, how many cats do you need to grow at home for your mom to go crazy, right? But peas, you can fit a bunch of pea plants in a small garden. And at the end of the experiment, your mother could just cook them, right? So that's what Mendel thought. Now he's ready. So he has different types of peas, right? He has yellow peas, green peas. He can cross these and observe the babies, and observe the F1 generation, see how they look. He could cross smooth peas with wrinkled peas. He can cross flowers of different colors, different pod shapes, different things. So now Mendel is ready, and he's gonna start with one of those 6 to 7 traits. So now he starts with the first trait. So he says, okay, he has a green peas plant and a yellow peas plant. He crosses them. Does he just go right away and crosses them right away? No, he did not. So before actually doing his experiment, he went ahead and crossed green with green. And every time he crossed green with green, he got green. And also he crossed yellow with yellow. And every time he crossed yellow with yellow, he got yellow. And this was very important. And I'll tell you what in a little bit. So then, and only then, did he do an experiment. So now that he's sure that these plants bred true, they are true lines, pure lines, or homozygous, as we'll learn later. So now he did his cross. So when he crossed green with yellow, in the F1, he got yellow. So, and he saw that the green kind of disappeared, and he called this recessive trait. So, and then the F1, so we call this the F1, which is the progeny, the first generation, was completely yellow, was as yellow as the dominant parent. And this was significant because at that time there was the competing theory of inheritance that said if you cross green with yellow, you'll get something intermediate, something that looks like this, right? But in Mendel's case, he got actually some that was yellow, 100% yellow as one of the parents, right? Not some type of intermediate trait. So And Mendel called this the particulate theory of inheritance. So the F1 was not an intermediate phenotype, it was yellow. But does this mean that competing theory of inheritance, they call it the blending theory, right? Is it completely wrong? Well, in Mendel's case, it's not the case, right? He didn't find the blending inheritance to be true. But we know in real life that this blending inheritance is also true. So Mendel's traits, it's either yellow or green, either or, right? In the case of humans, for example, some of the traits, you could have the earlobe either attached or not, right? So, and also your ability to roll your tongue, you could either do it or no, right? Like this, I can do it. So it's either or, there is no intermediate phenotype. But we know that the blending theory is also correct, right? So in the case of human skin color, for example, some of those traits, it's not like you're either white or black. Well, there's all shades and all colors in between. So let's go back to Mendel. So Mendel crosses two different colors of peas. The F1 or the first generation is completely 100% yellow. Then he said, well, let me go to the second generation. Let me take those peas plants and cross them by themselves, right? Let me cross yellow by yellow. And when he crosses them, what he finds is, can you guess what did he find? Peas. Yes, he found peas. So he found peas. And the green color actually reappeared again. So, and when he looked at the ratios, it was 3-1. 3 yellow to 1 green. So, and those are the actual numbers that Mendel has gotten, right? He got 6,022 yellow versus 2,001 of green. But this was not the only trait that Mendel has studied. He actually did a bunch of traits. So now that the seed color has worked fine for him, he said, okay, let me try these other traits. So he tried flower color, and he crossed seed shape, then he crossed pod color, green versus yellow. He tried different traits. And every time he did that, he got the F2 or the second generation was about 3-1, right? These are his actual numbers. So it's kind of close to 3-1. So these results at Mendel's time, they were kind of strange and unexpected. And whenever you come up with something unexpected, right, you have to offer some type of explanation. So Mendel wants to come up with some type of explanation. So then Mendel says, okay, here are the two parents that I have used. These are the two parents. I crossed them, and the F1 is yellow, right? So he said the reason for this is, because each parent has two factors, two letters, right? Little y, and this one has big Y, big Y. So, and Mendel called them factors. He did not know the word alleles, right? The word alleles was not invented yet, right? That will come up later. So those are two, he called them factors of inheritance. And he says two parents, and each parent passes one of these two factors to the offspring. So this one gets little y and big Y. So this ends up big Y, little y. And that big Y, little y, that individual looks yellow because he's dominant, because he says the big Y is dominant to the little y. So, and then when you cross this to get the second generation, you cross it with a similar individual, and that's what he gets. He got 3-1 yellow, right? So let's see how we got this, how we got to these. So let's do the Punnett square. So we have two individuals. So this is big Y, little y. This one is big Y, little y. So each one of those can give two types of gametes, right? So each one of those, these are gametes. So this can give big Y, little y, and this one big Y, little y. And when you, it gets this from mom, this from dad, you get big Y, big Y, little y, and big Y, little y, little y, little y. And this is the genetic composition, right? We also call it the genotype. So what color do these plants look like? Well, big Y is yellow, big Y, little y is yellow, yellow, and green, okay? So and that gives us this, these different different genotypes. So again, Mendel studied different traits, as we mentioned, and those are the actual numbers that he got. So let's talk about just summarize what we've talked about so far and give you a few definitions. A gene, right, in the case of peas or anything, a gene is a unit of heredity, right? A specific location on the chromosome that a parent has, or any individual. It's being passed from parent, that gene. And that gene is responsible for a specific trait, such as seed color. So in this case, our gene would be the seed color gene. And we have two versions of that. We have yellow and green. And these two versions, these are called alleles. So an allele is one of the two or more alternative forms of the gene. So in the case of the seed color, we have green and yellow. So green and yellow, those are or big Y and little y, those are the alleles, right? So the trait is the seed color. So the gene is a unit of heredity, and you have one or two or more variables of that specific gene or location. Okay? So we also, the word homozygous, it means pure. If you remember the P example, if you are big Y, or little y, right here, so this is pure, you call it homozygote or homozygous. This is homozygous. When you have two alleles that are identical, that's homozygous. If you have two alleles that are different, right, that's heterozygous. So let's go here. So homozygous is an individual having the same two alleles. A heterozygote would be an individual that has two different alleles. Okay? So a genotype, that's the genetic composition of an individual. So if you are big Y, little y, or big Y, that's your genotype. Yeah, that's the genetic composition. So if you are big Y, if that's your genotype, then you are yellow, right? And that's your phenotype, right? We call it phenotype. So I mean, we could have just have said phenotype, but scientists, we like to make things complicated, so people think we're sophisticated. So phenotype it means how it looks, right? The appearance. So in this case, you have green versus yellow. Okay, so what have we learned so far? Well, we've learned about Mendel's factors of inheritance. So that each parent carries two factors, big Y, big Y, little y, or little y. And these factors we know that they're now called alleles. So these alleles are passed from parents to offspring. So each parent passes one allele to each of the progeny, and then the offspring, right, the kids, they get one from each parent. And the phenotype, how an individual looks, whether it's green, yellow, whatever, it's determined by the genotype. So the genotype determines the phenotype. The genetic composition determines how the individual looks. And lastly is the dominance, and it's the ratio. So I've seen that when you cross two pure lines, homozygous individuals that differ in one trait, the first generation will be completely dominant and the recessive trait completely disappears. And then in the second generation, you would see 3-to-1 segregation. And this law was called Mendel's first law, right? Or law of segregation. That's Mendel's first law. And of course, Mendel didn't name it Mendel's first law, right? Scientists, they don't do that, right? It was named later. So now that Mendel has done all of these traits, he did seed shape, seed color, different things. He said, well, let me move to the next step. And instead of just studying one trait at a time, which we call also monohybrid, he said, well, why don't I study two traits at the same time, right? And make it complicated. So he said, let me do a dihybrid, right? Which is studying two traits at the same time. So, and instead of just doing yellow versus green, right? Seed color, he said, well, and in this case, he got yellow all the time. He said, well, what about we do something different? We do yellow, like seed color, and seed shape. So instead of yellow and green, he did yellow and smooth shape, and he crossed it by a green and wrinkled. And he observed the first generation, and he saw yellow and round. So Mendel now saying, okay, the yellow, we've already established that yellow is dominant to green, but now he has learned that smooth or round is actually dominant to wrinkled. So let's now cross this F1 with itself, cross it with the same individual, and see what we get. When he crossed these together, he got a Punnett square of 16 different genotypes. And to construct this table, what you would do is, you know that this big R, little r, big Y, little y. So we know the big Y, this is the yellow, the little y is the green. Similarly, he picked big R for the round and little r for the wrinkle. And we know that each individual can give 4 gametes. So this individual can give big R, big Y, big R, little y, little y, big Y, sorry, little r, big Y, or little r, little y. The same here, big R, big Y, big R, little y, little r, big Y, or little r, little y. You cross these, so you have to combine them together, this with this, this with this, and you would get 16 different genotypes. So let's see what do these look like. So now that this is the genotype, what is the phenotype? Well, these are yellow, yellow, green, green, and that's basically based on the genotype. So this is big R, so I know this is round, and big Y, little y, this is yellow. So, and if you look at this one, he has little r, little y, and that is wrinkled and green, right? Because when it comes to the green color, you have little y, that's green, little r, that is wrinkled. So we have now seen new combinations, right, that we didn't see before. So we had mom was yellow and round, and dad was green and wrinkled. But now we have new recombinations, right? Stuff that we haven't seen before. So we have green and round, we have yellow and wrinkled. So, and the ratio of that would be 9 to 3 to 3 to 1. 9, that is dominant for 2 traits. 1, that is recessive for 2 traits. 3 individuals dominant for 1 trait and recessive for the other. And 3 also dominant for 1 trait and recessive for the other. So, and this might seem complicated, but let's look at it and look at the seed color, right? And ignore the seed shape. So let's look at this, count the seed color. How many are yellow? 12. How many are green? 4. So 12, 12 to 4, that's 3 to 1, okay? Okay, so let's look at the seed shape, right? And can I ignore the color? So look at the seed shape. I have 12 round and I have 4 wrinkled. That's also 3 to 1, right? So it's the same rule applies. So he called this, he didn't call it, later it was called the law of independent assortment. So you have these 2 traits are being distributed and assorted independently. Okay. So let's summarize Mendel's work, or the second law. So, which he called, it was literally called the law of independent assortment. That when you have a dihybrid cross, you have an individual, 2 individuals that differ in 2 traits, right? And then, so, and Mendel says, okay, so when you have 2 crosses, like 2 individuals that differ in 2 traits, right? Green and yellow, and smooth, like round and wrinkled, right? Seed color and seed shape. Green versus yellow, and round versus wrinkled. That the F1, the first generation, you'll see 100% yellow for 2 traits, for the 2 traits. The second generation, it will segregate into 9 to 3 to 3 to 1. So, and that was all great work, and Mendel did a lot of cool things, but actually, nobody recognizes Mendel's work, right? So Mendel dies, and he rests in peace. But later, in the late 1800s, that's when Mendel's work would be rediscovered. And it actually, the answer came from the field of cytology. So, and cytology is like looking at the cells under the microscope, because microscopes were becoming better and better, especially in Germany. And then scientists, they came up with these different dyes. And they would put cells under the microscope and put different dyes and stains on them. And whenever they look under the microscope, they saw like these funny structures that would absorb the dye, right? Whenever they put a green dye, these funny structures would absorb the dye and get colored. When they put different types of dyes. So every time they put dyes and they look under the microscope, they would see these funny structures. and they didn't really know what they were, right? So they called them colored things or colored bodies. Chromo means color, somes meaning bodies. So they call them chromosomes. So the word chromosomes is not like any fancy word, right? It's like colored things because scientists they didn't have an idea what that was. So they call it colored things. Okay, but we still didn't know how Mendel's work was rediscovered. Well, when they looked at these chromosomes under the microscope, the way these microscopes moved and separated under the microscope looked like what Mendel was talking about. They looked at the cells that were undergoing mitosis, and I know that you guys already studied that. So the chromosomes would line up that way, right, at the central line of the cell. And when it's time to divide, this is what happens. So the cells, they will get separated. The two pairs of chromosomes will get separated. And then they would double, they would disappear, and reappear again. So now we have a new two cells, right, that each has the same number of chromosomes. So let's look at meiosis. So they were looking at cells undergoing meiosis, and they have noticed something different. Is that these cells, right, the chromosomes would line up this way. So, and will later then be divided into two cells. Now we could see that we have reduced the number of chromosomes. So now I have 4 different gametes or 4 different cells, right? Each one of those cells has half the original number of chromosomes. So and scientists were like, okay, so each cell has 2 factors and it's passing 1 factor to the progeny. So they said, well, this sounds like what Mendel was talking about, right? So these must be the alleles or the factors of inheritance. So they said these must be Mendel's factors, like each parent has 2 copies and they pass 1 copy to their offspring. So these chromosomes must be Mendel's factors. So let's look at the chromosomes and how they actually look. So if you have a chromosome, like a pair of chromosomes, these are the homologous chromosomes. Each chromosome has DNA. If you unpack that, you will find like double-stranded DNA. So if you consider that this one chromosome, right, imagine that this individual is big Y, little y, right? So if this chromosome or this gamete is coming from mom, so mom, or this coming from dad, so dad will have this specific sequence of DNA, for example, and mom would have that sequence of DNA, right? So you could see that this double strand coming from mom, double strand coming from dad, and they are different, right? They're almost identical except for that specific letter. So I can call this the Y allele, right? So this is the Y allele, the big Y allele, and this is the little y allele, okay? So an allele is one version of the gene, right? And it has a specific sequence of DNA, and this is the other allele, okay? So an individual that has these 2 big Y, little y will end up being yellow or green? Yellow, true, okay? So I hope that this was helpful and useful. So, and if you have any questions, please write in the comments and I'll make sure to respond to them. Thank you.